So in the last section, we looked at things that were strictly Mendelian genetics. They were either dominant or they were recessive. And when there was a dominant and a recessive in a heterozygote, the dominant covered up the recessive completely. In the next section, we're going to look at some variations. So these are exceptions to the rules that don't really follow exactly the rules that Mendel laid out. The first exception is called incomplete dominance. This happens when there is an individual that is heterozygous, meaning they have one dominant allele and one recessive allele, but instead of just showing the dominant trait, they show a blended or an intermediate form. So the quintessential example that your textbook gives is on the right. This shows the snapdragons. These are a plant. A homozygous dominant, big R, gives a red flower. And a homozygous recessive, little r, gives a white flower. So when these two true breeding plants are bred, we get an F1 generation. So the next generation is all hybrids, big R, little r. Only instead of being red, the red and the white blend to create a pink flower. Then if we do an F2 generation, where we have a cross between two pink flowers, we get an F2 generation, where we get one red flower, two pink flowers, and one white flower. So in this case, the pink is the intermediate or blended phenotype, and the F2 generation is going to have one that has the dominant phenotype, two that have the blended intermediate phenotype, and then one that has the recessive white phenotype. Another example of incomplete dominance is hypercholesteremia in humans. A normal cell on the left is dominant, which has many receptors for LDL cholesterols that helps remove cholesterol from your blood. A recessive individual, completely recessive, little h, has no receptors and has the inability to take cholesterol into the cell, which means that they end up with high levels of cholesterol in the blood, which causes severe damage and heart disease. An heterozygous individual, instead of having a blended, has kind of an intermediate. They don't have a lot of receptors, but they don't have no receptors. So this person would have a mild form of the disease where they can take in some cholesterol, but not very much. And so they do get some cholesterol building up in the blood. Another form of variant is called codominance. Codominance is when two genes that are both dominant get expressed equally. Instead of one covering the other up, or any kind of blending or joining, they both get expressed equally. One example of this is the ABO blood groupings. So ABO blood groupings have 3 alleles instead of 2. Big I-A and big I-B that are both dominant, and then little I, which gives the phenotype O, which is recessive. So let's look at the blood groups. On the right in the graph, if you have big IA and big IA, or big IA and little I, then you have blood type A. And if you look at the picture, here's a red blood cell, and it's got little triangles on the cell, and that's representing the antigen that is on the cell for gene A. If you have big IB, or big IB little I, then you have type B blood. And if you look at the picture, here's the red blood cell, only this has a different protein. This is coded for by type B, so this one's a circle instead of the triangles. So somebody who's heterozygous, who has big IA, big IB, is going to have type AB blood. And they express both A and B at the same rate. So on the red blood cell on the right, they have the triangles from gene A, and they have the circles from gene B. They're not blended together in any sort of intermediate. They're just both expressed equally. And then blood type O, little I, is the homozygous recessive, and it doesn't have any antigens on the blood. So that's what makes blood type O the universal donor, and blood type AB the universal recipient for blood transfusions. Another type of difference from Mendelian genetics is called pleiotropy. This happens when one single allele change or a mutation causes lots of different effects. So we're going to have a mutation in the genotype, and it's going to affect many different phenotypes. One example of this is called sickle cell disease. Sickle cell disease is a mutation in the red blood cell hemoglobin proteins, which causes the red blood cells, instead of them being discoid, like these red blood cells on the left, they look like these sickles, these little half moons. These half moons are pretty bad at carrying oxygen, and they tend to get stuck together in the circulatory system. So having this one mutation causes lots of different effects. It can cause damage to organs like your kidney and your heart and your brain, and it can cause other effects like joint problems, pain, physical weakness, anemia from not having enough oxygen in the blood, and it makes you susceptible to other sorts of opportunistic infections. So all of this, these bad effects down here are different phenotypes, but they all result from a single genotype change. So polygenic inheritance is kind of the opposite of the last one, pleiotropy. Poly means many. So polygenic inheritance is when many genes add up to result in one single phenotype. Some examples of this in human are skin color and height. So in the person on the right, we're looking at 3 different genes, A, B, and C, that all code for skin color. So in the F1 generation, you can see that 2 hybrids were crossed, 2 people with moderate heterozygous traits for all 3 genes, and they have a medium shade to their skin color. And you can see the giant Punnett square down here. Results in this huge variety of options of phenotypes, from very light skin tone to a very dark skin tone. The graph on the left shows a distribution, where most often you're going to have individuals with a moderate to average skin tone, a kind of an average of their parents. But it's very possible to get a child with who's got lighter skin than the parents or darker skin than the parents. And this is because in real life, there's much more than 3 genes that code for how much skin protein, melanin, which is the color in your skin, that you actually produce. And so an offspring can have a very different skin color than their parents based on which genes they inherit. The same thing's true for height, human height. Most people are of an average height, but there are some people that can be shorter or taller than their parents based on which genes they inherit. The last thing to think about is how the environment affects our genes. The environment tells our genes how to be expressed, which can cause variations in phenotype, even when there's no differences in the genes, the genotype. So think about exposure to sun. Your skin is probably one color, and when you go out in the sun, it changes color. Maybe if you get sunburned, it becomes red. If you tan, it becomes darker. So your genetics didn't change. Your genes are still the same. But what you changed was your environment, the amount of sun that you were exposed to. Nutrition has been shown to have a huge effect on our genes. Things like meat and dairy can actually affect the genes and turn on cancer genes. Antioxidants that you find in like blueberries and other types of berries have been known to be very protective against damaging DNA. Exposure to drugs, tobacco, smoke, alcohol, all sorts of things can affect how our genes are expressed. If you look at the picture on the bottom right, these are identical twins. They look very similar, but they have completely different hair colors. One of them has kind of a reddish blonde, and then the other one has black hair. Their genes are the same. They're identical twins, but something in their environment, something outside of their DNA, changed how they were affected. There are studies that have been done on twins where one twin is found to be right-handed and one twin is left-handed. And it's just a difference of which genes are on and which genes are off.